As millions of tons of plastic waste degrade into fraction of a micron particles, capturing them becomes a severe physical chemistry problem. Traditional mechanical water filtration operates using physical barriers. When mesh sizes are reduced enough to trap submicron particles, the resistance spikes, fluid flow grinds to a halt, and the systems rapidly clog. The industrial alternative to physical barriers is chemical flocculation, introducing compounds that cause particles to clump together for easy removal. But microplastics are highly nonpolar. In natural environments, they physically repel polar water molecules and standard chemical binders. The carbon backbone of plastics makes their surface chemistry inherently stable and highly hydrophobic. They resist attachment at the molecular level. Any scalable solution to microplastic pollution must bypass this resistance, or actively weaponize that nonpolar surface chemistry against the plastic itself. Bioremediation has attempted to solve this using extracellular polymeric substances, or EPS. These are naturally sticky compounds secreted by microbes to trap suspended particles. The effectiveness of EPS relies entirely on the specific species of bacteria, and the kinetic rate of capture is incredibly slow. It routinely takes over 6 hours for EPS-secreting bacteria to achieve even modest microplastic aggregation. A recent study published in Nature Communications bypasses the EPS mechanism entirely. The researchers developed a platform called RUMBA, the Remediation and Upcycling of Microplastics by Algae. The biological workhorse driving this system is the Cyanobacterium Cyanococcus elongatus, specifically a rapid-growing strain known as Utex 2973. The RUMBA protocol relies on 3 simultaneous processes. First, genetic engineering maximizes surface hydrophobicity. Second, feeding on municipal wastewater. And third, upcycling waste into bioplastics. By altering the physical chemistry of the cell surface, the RUMBA platform shifts microplastic capture from an inefficient disposal problem into a continuous manufacturing loop. The core modification to the cyanobacteria involves inserting genetic pathways that force the cell to produce and secrete a compound called limonene. Limonene is known as a highly hydrophobic molecule. As the cell produces it, the compound accumulates and enriches on the outer surface before it has a chance to evaporate into the environment. This surface coating physically alters the organism, creating what researchers classify as hydrophobic cyanobacterial cells. Wild-type cyanobacteria possess a highly negative charge in their outer layer. This negative electrostatic field naturally repels suspended plastics. Limonene secretion suppresses that charge. The zeta potential drops from negative 25.78 millivolts in wild-type cells to negative 13.91 millivolts in the HCC strain. Reducing this negative surface charge weakens the electrostatic repulsion. This exact thermodynamic shift allows the innate hydrophobic attraction of both the limonene and the plastics to take over, causing them to rapidly cluster. Operating with this engineered surface chemistry, the HCC strain removes 91.4% of polystyrene microplastics from a solution in exactly 1 hour. Transmission electron microscopy reveals the aggregation. Top row, notice the sparse attachment of polystyrene to wild-type cells. Middle row, massive clusters of plastic trapped where limonene accumulates. Bottom row, stimulated Raman scattering confirms the mechanics. The green limonene signal directly overlays the red polystyrene. This spectral overlap proves the limonene acts as a localized molecular glue, driving a physical aggregation rate that standard biological pathways cannot replicate. To verify that hydrophobicity is the sole mechanism driving this process, researchers introduced a surfactant called tween-20 into the clustered suspension. Surfactants operate by lowering surface tension and physically disrupting hydrophobic links between molecules. Immediately upon adding the surfactant, the microplastics and cyanobacteria cells decoupled and resuspended in the fluid, confirming that hydrophobic attraction is the specific physical driver of the capture. Relying on a physical force rather than specific biological receptors means the capture mechanism is agnostic to the type of plastic present in the water. Laboratory trials confirmed the modified cyanobacteria successfully aggregate both polyethylene terephthalate and standard polyethylene particles. By weaponizing broad-spectrum physical chemistry, this system targets the complex, mixed polymer reality of environmental pollution rather than isolating a single chemical variant. To test the platform's viability out in the field, The process moved from controlled laboratory media into samples of raw surface water and municipal wastewater. Scaling this microplastic aggregation requires massive cellular growth, a metabolic process that consumes large amounts of nitrogen and phosphorus. The researchers deployed the engineered HCC strain directly into raw influent and effluent tanks at a municipal wastewater treatment plant. Over a 5-day period, the cyanobacteria extracted the required nutrients to grow, successfully removing up to 99% of the nitrate and ammonia from the wastewater. This intense metabolic extraction occurred concurrently with the microplastic capture. The presence of the plastics caused zero growth inhibition in the cyanobacteria population. Processing municipal wastewater handles a costly infrastructure requirement, while simultaneously generating the exact biological biomass needed to extract microplastics, effectively subsidizing the economics of the entire system. Current filtration technologies suffer from a critical logistical bottleneck. Capturing the microplastics generates a secondary, highly concentrated hazardous waste sludge that requires permanent physical storage. The RUMBA platform eliminates this secondary waste entirely. Looking at this process flowchart, the co-sediment of cyanobacterial biomass and trapped microplastics is extracted and cast directly into this golden-green composite film. Inside this new film, the aggregated cyanobacteria cells act as an integrated biopolymer filler embedded directly within the captured microplastic matrix. Graphing the mechanical properties in Section E shows the biological filler drops baseline tensile strength. However, Sections F and G reveal the biopolymer triggers a 2.3 times increase in elongation and a 2.2 times increase in toughness compared to pure plastic. The process circumvents the generation of a toxic waste stream by upgrading the mechanical tolerances of the recovered polymer, yielding a durable, high-value composite material. Scaling this cycle to industrial output requires evaluating the commercial viability of open pond cultivation systems. The techno-economic analysis calculates a minimum selling price, or MSP, of $3.58 per kilogram of upcycled bioplastic produced by the pond system. At $3.58 per kilogram, this composite material successfully competes with standard commercial bioplastics like PHA already operating in the market. Moving to the life cycle analysis, researchers tracked the total environmental footprint of operating this process at scale. By powering the system with renewable solar energy and factoring in the cyanobacteria's natural consumption of carbon dioxide, the operation achieves a net negative emission of 4.5 kilograms of CO2 per kilogram of bioplastic produced. Aligning cellular surface engineering with downstream polymer science transforms microplastic remediation. The RUMBA platform takes a permanent environmental hazard and turns it into a profitable, carbon-negative manufacturing feedstock.